Welcome to all. Myself Dr. Anju Sharma. Today we are talking about plant tissue culture technique. As we all see that plants are grown in soil and field condition, but by plant tissue culture technique we can grow the plant in laboratory. How it is possible? Let's see. Firstly, we go for basics of plant tissue culture. Plant tissue culture is the technique of growing plant cell, tissue, or organ in an artificially prepared liquid or solid medium under aseptic condition. The technique of plant tissue culture is based on the concept totipotency. And what is totipotency? Totipotency is the ability of a single cell, organ, or any tissue to generate whole plant. The idea of totipotency of plant cell was first given by Gottlieb Haberland in 1902, and he is known as father of plant tissue culture. He is the first person who cultured the isolated vegetative cell on simple nutrient medium. Basic requirement of plant tissue culture. These are some tools and chemicals present in our laboratory very usually. And in first slide, we used the term aseptic condition, means germ-free condition. For that purpose, we use these two instruments. First is hot air oven. Hot air oven is an electrical device which used dry heat for sterilization, and in this apparatus we uh, sterilize the glassware and our tools. The second sterilization apparatus is autoclave, which works on principle steam under pressure, and the moisture of steam kill the all infectant present in nutrient medium. The third important apparatus is laminar airflow cabinet. It consists UV light and HEPA filter, high efficiency particulate air filter. The device works by the use of inward flow of air through one or more HEPA filter to create a particulate free environment, and the whole inoculation work done on this cabinet. This we all know, this is pH meter, which is used to set the pH of our nutrient medium, that is 5.8 to 6. Nutrient medium or culture medium. This is very important because plants are grown on this medium in laboratory. A liquid or solid substances containing nutrient in which any plant cell, tissue, organ cultivated for scientific purpose is known as nutrient medium. It contains inorganic salt, both macro and micro elements, organic nutrient. Usually we use sucrose, vitamins and amino acids. Several media were used for plant tissue culture. These are white medium, MS medium, B5 medium and 6 medium. Among these medium, the most popular medium which is used for plant tissue culture is MS medium, which was given by Murashige and Isku in 1962. With this basic medium, various growth regulators like auxin, cytokinin, and gibberellin were also added as per required. These are some related terms which we used in plant tissue culture technique. We will discuss further. Now the step of plant tissue culture. First is the selection of plant. This is very important step. जिस प्लांट को हम सेलेक्ट कर रहे हैं वह प्लांट डिजीज फ्री होना चाहिए और एलीट होना चाहिए. एलीट मीन्स आइडियल होना चाहिए. आइसोलेशन ऑफ एक्स प्लांट. सिलेक्शन ऑफ प्लांट के बाद में जो नेक्स्ट स्टेप है वो एक्स प्लांट को आइसोलेट करना है. एक्स प्लांट इज द प्लांट पार्ट विच इनिशिएट द कल्चर. Next step is sterilization of X plant. Sterilization of X plant, which is done by mercury chloride or sodium hypochloride. Inoculation of X plant. Incubation of cultures. When inoculation done, we kept the cultures in culture medium with appropriate temperature, light, and humidity control condition. After incubation, after 4 to 5 weeks, plant respond fully via direct pathway or indirect pathway. We observe it carefully and we subculture the cultures for say for maintenance on same medium or further rooting medium. After rooted After rooting, rooted plantlets are hardened and acclimatized. Finally, they transferred into the pot. Regeneration pathway of plant tissue culture. There is two pathways for regeneration of flex plant. First is direct pathway or micropropagation. Here you can see when we inoculate the explant on culture medium, explant convert into multiple shoots, and these shoots finally rooted and uh, transferred to field. This is the direct pathway. Second pathway is indirect pathway. In indirect pathway, explant converted into callus, that is parenchymatous tissues, mass of parenchymatous tissues. Then after redifferentiation, this callus is converted into young plantlets, and finally they also transferred into pot. Now, the advantages of tissue culture. The new plantlet can be grown in short amount of time. Only a small amount of initial plant is required. The new plantlets and plants are more likely to be free of disease or virus free. The process is not dependent on the season and can be done throughout the year. The technique is especially useful for slow growing and endangered plant, and it is very effective method for commercial production of plant on a large scale. These are some questions which by solving these questions you can self-assess yourself. These are some resources. If you want to learn more about this topic, you can go through these books, journals, and suggested website links. Thanks.